So hello everyone, and welcome back to Simcyp Technical webinar. My name is Xian Pan. I'm part of the Translational DMPK group at Simcyp, and I'll host the session today. So before we start the talk, I'd like to kindly request that everyone stays muted during the presentation. Should you have any questions, feel free to raise your hand or post your queries in the chat box. We will have a Q&A session at the end to address them all. We're also recording the presentation, which will be available on YouTube shortly. Next slide, please. And it's my pleasure to introduce today's speakers, James Craig and Natalie Morris. James is a staff software engineer in Satara Data Science with over 15 years experience developing scientific applications. He's also the maintainer of the Satara R pharmacometrics package. Natalie is a research scientist at Simcyp with academic background in engineering, mathematics. She worked on various projects, including virtual bioequivalent, long-acting injectables, and also R scripting. And she led the development of the VBE workflow for the Paliperidone Palmitate. I hope I pronounced it not too wrong. So it's a project funded by a FDA grant, which will be focus of today's case example. And I'm delighted to have both of them with us today. In this session, they will walk us through a PBPK-based virtual bioequivalent workflow, showcase the integration of the Pirana and Simcyp, along with advanced statistical approaches to support formulation, evaluation, and study design. Without further ado, I will hand it over to Natalie. Over to you.
Great, thank you. Thanks. All. Okay, yeah, thanks for the introduction. We're going to talk for about 5-10 minutes on sort of background of Pirana and what the module can do, and then I'll pass over to James, who will give you a demonstration of the software. So just to start with the information on the funding. So the research here was funded by the FDA Office of Generic Drugs under this grant number here. And just so as a disclaimer, the views expressed here are not official policies of the FDA or the HHS and do not imply endorsement by the US government. So what is the Pirana Simcyp VBE module? So this is a new module within the Pirana workbench, which gives you an integrated tool for PBPK-based VBE assessments. It supports the automated generation of VBE trial simulations and also their evaluation through a series of inbuilt R scripts, which are in the workbench just here. To use this module, you will need a Simcyp license, a Pirana license, so for the latest release 25.7, and also the Simcyp long-acting injectable or LAI module license. Currently, we're just supporting VBE assessments with LAI formulations. We are looking to expand the compatibility to different routes of administration, but initially it's just for these LAIs. And the module is provided with a case study on 3-month LAI suspensions of Paliperidone Palmitate, which is the case study we'll talk about today. So what are the key features of the module? So it provides an integrated workflow which seamlessly links Pirana and the Simcyp simulator via the Simcyp R package. Also provides tools for sensitivity analysis in a Shiny app and also within R scripts. It allows you to conduct population simulations. So we have an accompanying Shiny app, which allows you to run population simulations. And essentially that uses Simcyp R in the back end to remotely call the Simcyp simulator, generates PBPK trial data, and then send that to Pirana, where you can then do processing of that information and your bioequivalence test. And the module also has a shareable project format, so work is contained in these VB project files that can easily be shared between colleagues for collaboration. And we also provide a range of built-in statistical tests and analysis scripts, which you can use for bioequivalence evaluation and visualization. One of the key kind of benefits of this module is that all of these R scripts can be user modified. So if you want to customize your analysis, customize the look of outputs, or expand the analysis in different ways, you can do that through just modifying these R scripts. So a bit of background on the case study that we'll look at. So again, this was developed as part of our FDA research project here, and it was looking at 3 months to LAI suspension formulations of Paliperidone Palmitate. So all of the simulations were using parallel trial designs. So also we currently are only supporting parallel virtual bioequivalence trials. We are looking to expand to crossover, but initially it's just been set up for parallel designs. The CQA we were interested in was the initial mean drug particle size, and in terms of how we set up the model, so we did this in this main simulator previously, but we essentially just fit a reference model to virtual patient data that we generated with a population PK model, as we didn't have any prior clinical data. And we did have some formulation data for the reference drug, and then to basically make the test formulation workspaces, it was just a case of changing those CQA values. So the VBE assessments that we did here were kind of sort of a speculative sort of look through CQA space to see how particle size was affecting bioequivalence decisions. So this is just an overview of the analysis tools that we've got available within Pirana. So we use a Shiny app for trial simulations and initial sensitivity analysis work. And then we have a library of R scripts that you can use for the automated VBE assessments and visualizations. And as I mentioned, again, these are all user modifiable and extensible. So there's 4 kind of key parts of this workflow. We have a script for power assessment, so looking at the probability of declaring equivalence for trials of different sizes. This is really useful for looking at your sample size assessments. We have a CQA sensitivity script, a type 1 error calculation script, and also safe space calculation. So I'll give a very quick kind of overview of these 4 components, then I'll pass on to James to kind of give you a full walkthrough. So for the power assessment, we calculate this in the module using a bootstrap Monte Carlo resampling algorithm. So essentially the sort of idea is that we generate very large pools of patients, and then in this bootstrap sampling algorithm, it generates trials of various sizes randomly from this overall pool, determines bioequivalence for each trial using the TOS test, and then we just work out power as the observed rate of passing trials. So if we simulate 100 trials and 50 pass, you have 50% power. So in this way, there's no analytic approximations of power, and it's all through actual PBPK simulations. And so this is an example output, and then the noise that you see underneath is just that Monte Carlo sampling variability. We have a CQA sensitivity analysis tool. So in this, what we're doing is we basically regress test formulation CQA values against their corresponding test to reference GMR. So this is specifically looking at point estimates of the GMR. And we regress it with 4 different versions of just a simple linear regression model, and we identify the best fitting model using the sum of the residuals. And the reason why we do this is because once we've got a reasonably good fitting regression model, we can use that model to predict the CQA values where you would expect a GMR of 0.8 or 1.25. So you can basically predict the CQA values that would be on the very edges of your bioequivalence region. And that directly feeds into type 1 error calculation. So type 1 error is essentially, it's a false positive rate. It's the probability that you are incorrectly determining that an inequivalent product is equivalent. So it's a false positive rate. And we calculate this at the edges of the BE region. So again, at GMRs 0.8 and 1.25. So again, we just find those CQA values using the regression step that we showed on the last slide. And then for type 1 error calculation, it's a similar script really to the power assessment, and it's basically just looking at the probability of declaring bioequivalence. And what we were looking for here was just to see that type 1 error was not significantly exceeding the 5% significance level of the TOS test. So just checking that type 1 error was being appropriately controlled through this VBE assessment. And then the last step in the sort of core workflow was safe space. So within Pirana, we calculate the safe space region for a given trial size and also for a given power threshold. The user can specify what trial size and what power threshold they want to look at. So you might start at 80, but you might want to also see what it looks like at 85%. You can change it and sort of modify the scripts in that way. And basically the output looks like this. So you've got on the x-axis, you've got your CQA values, and on the y, we're just showing power at that trial size of 200 subjects per arm. And the green region in the middle would be your safe space, because that is the region of CQA space where you're getting a bioequivalent result and your power is an acceptable level of over 80%. So it's sort of graded there by the trial design and the power threshold. So that was sort of a quick whistle-stop tour through the sort of stats elements of the Pirana module. And now I'll pass on to James, who will give you a walkthrough of the tools.
Thanks, Natalie. Go ahead and share my screen here. Just everyone can see my screen.
Yeah.
All right, so what we're looking at here is the Pirana interface. We're in the VBE run mode. So for those who are familiar with Pirana, you may know that Pirana sort of originated as a workbench for non-mem, where you would be in the mod run mode here, which is your extension for your non-mem control file. We've since made Pirana compatible with NLME in the metamodel context. So again, for POP-PK modeling workflows, Darwin for machine learning, automated POP-PK model development, and recently VBE for the Simcyp virtual bioequivalence integration. So now that we're in this VBE run mode, let's go ahead and point out some components of this workspace. So here in this Files tab, right, you can see all the files that are in this workspace folder. Again, Pirana is really just a workbench that overlays directly on your file system. So you could see this folder that I'm in, and then you could see the local folder in my File Explorer. And we have these VBE objects in here that Natalie mentioned, and we'll go ahead and take a better look at these here in a second. All right, so where do you get started? Well, I have my Simcyp workspace file in my working directory. I have my observed data, my XML file. And then it's just simply going to models, new model. You can select the locations of those files. I've already selected them here. And then you would just designate, all right, is this virtual bioequivalence simulation a reference or a test group? So you would make that determination here, and then you would hit create. So I've actually already pre-populated a few simulations here. When I hit that create button, well, what happens is the Shiny application will appear in your web browser. And there's quite a few inputs in here, certainly not all the inputs that you would have available in the Simcyp simulator, but we've made a subset of inputs available here for the virtual bioequivalence analysis. So the input that we're using for the Paliperidone case study is the particle size distribution, log normal distribution, the radius mean here, set at a value of 4.6357. So I've directly set this value in my Simcyp workspace. Every other value, right, that isn't populated here, essentially is just going to be used from the values set in the Simcyp workspace. So here I'm in the Simcyp workspace, again, where I would access that reference value here in the workspace is in the substrate, absorption, formulation index, particle radius, polydispersed, and it's this radius mean value that we're going to vary in the Shiny application for our virtual bioequivalence. All right, so let's jump back to the Shiny application here. So the first step would be to generate some simulation output for your reference group. So I've done that here. I've set this value of 4.6357. I've gone to simulate. I've hit the simulate button. I've selected this group, 4.6357, and I've selected run. I have the results here from the virtual population simulation. And now I can just simply save and exit, return back to Pirana, and you can see that for this VBE 17 group, it was the reference group. We could see the corresponding workspace and XML file, and then that CQA value that I used for the simulations here, 4.6357. So Pirana knows what value that I inputted in that Shiny application. And then there was only 10 subjects that I simulated from. So you can see the number of IDs here and the total number of observations. So I would do that first for the reference, right? So our 4.6357. Next, I would simulate for the test group. So I actually have some test group here, VBE 18, that I created. Now, before we actually simulate virtual population for the test group, something that is useful in this application is sensitivity analysis that you can perform here in the GUI. So any of these, the inputs, as you can see here, I can set one value or just multiple values here as a comma-separated string. And if I go to my simulate tab here, you can see that these parameters have changed, right? Now I have this 5, 6, and 8. 3 groups which to simulate from. These inputs also support sequence statements. So if I wanted to perform some sensitivity analysis, using a range of CQA values from 2 through 8, incremented by 1. Right, that's supported here in this application. If we go back to our parameter preview, you can see that these values have indeed been updated here. So if we were to conduct sensitivity analysis next, right, you can see all the unique parameter combinations that have been enumerated in the application. Again, I've only selected one CQA value here, but we can enter multiple values, and every single unique combination of those parameters would be expanded here in this grid, which we can use to perform a type of sensitivity analysis. Now, this sensitivity analysis is essentially a simulation for a representative subject of that population, of the virtual population. So I've already ran the sensitivity analysis. Go to my results. You can see the sensitivity analysis here. I can select from these different iterations and sort of preview them here in the plot, see how they fit the data, for example. And once I've, kind of selected a range and previewed various parameters for the population representative, the next thing to do would be to carry over the simulation for the virtual population, the entire virtual population, and a set number of subjects. So that's what I'll do next. I'll hit this simulate button, and I'm going to run a virtual population simulation. We can parallelize the runs. It will run about 30% faster, but there are some diminishing returns. Essentially, if you're using a small number of subjects, it's often more efficient to run it not in parallel. But keep in mind, if it is parallelized, it does consume a lot of resources on your machine. So what I'm doing here is identifying that parameter value of 6, right, that I selected here, and then the CQA that I'll be using for this virtual population simulation for the test group, and then I can hit run. So next thing I'll do is navigate to the virtual population tab. So it says it's running virtual population simulation for the test group, 10 subjects. This will take about another 10-20 seconds to finish. And I'll point out here that here in our documentation, which is available in the slide links, you can see kind of the different CQA values that were simulated here and the corresponding run times as well, estimated run times for the given number of subjects. All right, our simulation for this test group has completed. I'm going to go ahead and save and exit. You also have some options here to control the plot, outputs, mean concentration, interactivity, etc. All right, so what I just did there, go back to Pirana, refresh. You can see I have 2 VBE simulation objects I created here. So one's this reference group with a CQA of 4.6357. One's the test group with a CQA of 6 here. So now you can leverage the R scripting capability to perform some statistical analysis on the results. So I'll go to the scripts tab. What if I just want to plot the time concentration profile of both of these? I can multi-select them, concentration time predictions. Oops. Just double click this script. You can see we have various input options that are available. We'll take a look at those in a second, but it's just as simple as selecting your VBE and then run script. You can see here those 2 VBE plots. There's a time filter that I had set here in the data. Now, all this is completely customizable and extensible. So all these scripts, for example, if I wanted to make some changes to these, let's just say I can right-click the script, edit script. You can see that these scripts have these arguments. Now, this is the template script, right? There's a single time filter value here. What if so that's for the upper range, so this will filter values out that are less than this time filter value. If I wanted to have some like interval, for example, it's just as simple as copying one of these existing arguments, changing the tag low, for example, time filter value low. And then you just need to do something with the values that the inputs in the corresponding script. below. So you can see here, this argument is time filter val. So let's see where that is in the data in our script. You can see that I'm using it here just as a filter, right? So I would add that additional filter if we wanted an interval rather than just a limit here. And then I would just simply file, save as. Now I can save this script in my Pirana project folder. Go to demo projects, VBE analysis. Now any folder in Pirana that is named Pirana scripts located in your Pirana working directory. I'm going to just call this new. You can see that I've made a change to this script, copied it into a location, and I can go to my project now from the scripts tab and see that I have a custom version of this script that I can use, right? I made some changes, and now I can use that for any custom objectives, right? So now you can see that I have this additional filter time value, and I can put this as like a lower upper type of a range if I wanted to, for example. So this is just kind of a contrived example, simple filtering customizations. But when we look at some of the more complex statistical analysis scripts, we'll see how these customizations can be even more powerful. So I'm going to go back to the script library. Again, the project library is your customized versions of scripts. The library is sort of the template version of the scripts that is shipped with the Pirana installation. And it's really just as simple as selecting your VBE outputs, right? So now we're going to look at some of the simulation results that are contained with the Paliperidone example, not these simple 10 subject simulations that I just ran. And these are all available for publicly accessible for those who are licensing the Pirana Simcyp VBE module, so you can completely reproduce these analysis results. So if I wanted to perform the power analysis, well, it's just a matter of selecting various VBEs of interest here. So I'll select my main reference VBE that I've simulated. There's 100,000 subjects in this simulation. I can select different other VBE objects, my test VBE objects here. So we'll select all these ones that I've labeled here as power curve. And this is all customizable in Pirana in terms of the description. You can add different notes and info that just helps you kind of organize your outputs in terms of what VBE objects you're wanting to use for downstream statistical analysis. So with these VBE objects selected, well, it's just as simple as double-clicking the script and selecting a run script. Now, this script takes the longest to run, probably. It depends on the number of subjects, but for this particular example, it takes about 5 to 10 minutes to generate all the power results. Thankfully, those are always cached every time you run them inside your Pirana working directory. You can see that we have this Pirana temp folder. So all those power results, right? Here's the VBE reference is the first VBE, and then the second VBE is the test, and then the corresponding power. So once you've generated those, well, those results actually feed downstream to the other scripts. So now that we have power, for example, I can just simply double click this script and calculate the power for a given trial size. Maybe for the trial size set at 200 here, just select run script. So here you sort of get this power curve across the different CQA values. Now, if I wanted to set a different trial size, well, I can just double click that script, rerun it, maybe set this to 250 now, run script. And then now you can see the curve with a large trial size here at 250 compared to trial size of 200. Now, all these results, I'll just briefly run another script here, maybe the safe space analysis, just so we can see what that looks like. I'll do that for all my VBE, just so we can get a full curve here. Run this script. And here's the power curve that we saw in the presentation deck. And the important thing is all this is traceable, right? So any script in Pirana, you can also open directly in RStudio. And you can see the actual underlying script that gets transformed from that template script into a completely executable script here. So here are those arguments. Here are the values that Pirana transfers into this list called arg. Here's all those dynamic inputs that we selected in the interface get transferred into this script. And they have all the file locations here, as you can see, for the different reference and test files. And then downstream is just the statistical analysis and the different plotting code to create your plot outputs. Here we're creating a ggplot. So again, all this is customizable, traceable, reproducible, and very extensible in the context of Pirana. All right, I think we're at time here. Let me just close with few housekeeping items here, pointing out our publications. So here's a location of the recent publication for the Pirana workbench, including forthcoming papers on the Paliperidone Palmitate case study. Where to get Pirana? So for those who don't have access to Pirana, first step would be to go to my.certara.net and create an account if you don't have one already. You'll then see Pirana product there. You can select contact us and it will forward to our support and will facilitate access. If you already have access to mycertara.net, which is our CAD, Certara admin license authorization, it's just as simple as requesting access for Pirana. So you can see all the different products that your organization has purchased here, and then you can just select the request access button. And if you need any help, please email support@certara.com. And this link here at the bottom is the location of the Paliperidone example that we just demonstrated. There's a long form documentation. I'll point out that the next Simcyp webinar will be announced shortly. Registration will be available soon. Please visit this link to learn more.
Thank you.
And we'll have time for a few questions. I didn't leave much, sorry.
Thank you, James and Natalie. Yeah, we are open for the Q&A session. So if you got any questions, you can either raise your hand or type the question in the chat box. So I will just give it a go. I have a question about, could you highlight how the Pirana VBE module different from the Simcyp ones? Well, probably in another word, in what scenarios should we opt for the Pirana Simcyp integration? Probably the question for both of you.
Yeah.
Go ahead, Natalie.
Yeah. Thanks. Yeah. So for the so essentially there's some differences in terms of that sort of back end between the two. So as we mentioned with the Pirana module, it's calculated things like power from sort of long range estimates from your PBPK simulations. In the main VBE module, it's using analytical, empirical approximations of power and things like that, which is quicker to run, but in some circumstances it might not be as accurate as getting the actual exact estimate from these long range simulations. Pirana is also, it's meant to be something that where you can run sort of your simulations at scale and process them and sort of do that iterative reprocessing, because the data is all sort of stored in the workbench for you to kind of sort of reuse and work with. So there's benefits from that angle as well, sort of formulation design perspectives. But also there are also the caveats of we don't currently have all of the same trial design options and formulation types that you do have in the VBE module. The VBE module is, it's all sort of GUI based. You don't have to sort of you won't have to go into any scripts or anything like that. And it's all kind of preset to give your analysis out with clicks of the button. But if you do want more control over sort of the stats that you're doing and more control of the outputs and the analysis, Pirana would be better suited to that. And it will allow you to kind of go to scale and upscale to give you sort of views of safe space, which have been sort of also graded by power and things like that. So yeah, the stats are a bit different, and also it's can go to larger scales. Really, it's more customizable than than Simcyp. So it depends on sort of preferences and what questions you might want to ask with your VBE assessment, really.
Thank you. James, do you have anything to add to that?
Yeah, I mean, extensibility, customizability, that's, Pirana really gives you this window into the open source world of R, right? So all these results here in Pirana are just CSV, time concentration, right, in long form. So very easy to manipulate. All the data is right at your fingertips, and you can essentially build whatever workflow you want into Pirana, whether it be your own project-specific workflow or even organizational kind of standards and conventions that you also want to build out into your Pirana kind of reproducible workflows.
Great, thank you. I haven't seen any further questions, so I guess it's time we can conclude today's webinar. Thanks, Natalie and James, for the great talk today, and thank you all for joining us. Should you have any additional queries later on, please don't hesitate to get in touch with us. So we are currently planning a series of technical webinars for next year, and further detail will be available soon in the members area. Thank you again, and we look forward to welcoming you to our future events. Wishing you all a lovely holiday season, and goodbye for now. Thank you.